modulate resistance of host to parasitic infection or infestation. Resistance of host to parasitic infection or infestation. Learning objectives. This module will introduce the student to parasitic immunity, factor that influence the host immunity, natural and acquired resistance to parasitic infection, type of parasitic immunity, etc. Introduction. What is resistance? Resistance is the defense mechanism exhibited by the host against a foreign body or a pathogen. The host is defense mechanism which is directed against the parasite, either kill or retardates their establishment and limit their multiplication. A host is said to be susceptible if it is capable of being infected by a specific parasite. This implies that the physiological state of the host is such that a parasite is capable of establish itself in the host. A host is said that be resistant if its physiological state prevents the establishment and survival of a parasite. Factor that influence host resistance. Factor that influence host resistance. Species resistance. Host animals are susceptible to some parasite and resistant to other. For example, majority of the helminths of cattle are incapable of infecting sheep and goat. The normal resistance of various species to various pathogens is due to presence of antibodies on their erythrocyte called isohemagglutinin. Breed resistant. Certain breed of animals show natural or innate resistance to some parasitic infection. Example: West African hump-lace cattle and Dama and Mutura are resistant to trypanosomiasis, and they are called a trypano-tolerant breed. Similarly, red Maasai sheep of East Africa is more resistant to Haemonchus contractus. Zebu bos indicus show high resistance to Bupillus ticks than exotic cattle Bos torsus. This is probably a result of selection of the most resistant animal over many years. Age of the host. Age of the host. Young animals are generally more susceptible than adult to parasite infection. Example: pig less than 4 months old are more susceptible to ascarism. Similarly, lambs are more susceptible to Monizia species tapeworm and Trichostrongylus roundworm. Conversely, young animals are more resistant to babesiosis than older cattle, and this phenomena is known as inverse or reverse age resistance. Nutritional status of the host. Poorly fed animals are more susceptible to parasitic infection. Example: vitamin A D B complex and iron deficiency may interfere with establishment and maintenance of host resistant to blood-sucking parasite. Genetic constituent of host. The best analyzed example of genetically determined resistance is sickle cell anemia and its role in resistance to malaria in humans. Humans who inherit the sickle cell anemia trait possesses hemoglobin S HbS, which confirms resistance on human to malaria. Sheep with hemoglobin A show superior resistance to infection with Haemonchus contractus and Ostratigia circumniscata as compared to sheep with hemoglobin B. Sheep with hemoglobin A mount an infective self-cure phenomena that sheep without hemoglobin A. Type of parasitic resistance. Type of parasitic resistance: innate resistance or natural resistance to parasite. Innate resistance or natural resistance to parasite is the resistance or immunity possessed by the host animal from birth. In other words, innate resistance is physiological incompatibility between certain parasite and the host that prevent the entry and establishment of the parasite in the host. Example, helminths such as hookworm and schistosomas may not be able to penetrate the skin and gain entry into the host. Trypanosomes, upon reaching the bloodstream, may be destroyed by substance present in serum. Intracellular parasites such as Theileria annulata of cattle may be unable to enter other host because of deficiency of essential surface molecules in the host cell of unnatural host. Innate resistance or immunity can be complete or absolute or incomplete or relative. A host is said to possess complete innate resistance or immunity when a parasite is not able to infect a host under any circumstance. Example, sheep cannot be infected with ascarid when a host innately resistant to a species of parasite become infected owing to lowered health. It is incomplete innate resistance. Acquired resistance or immunity is resistance acquired by the host during its lifetime as a result of exposure to a particular parasite or through inoculation of parasite antigen. Acquired resistance may be either active or passive. Active acquired resistance is resistance acquired in response to the introduction of live or killed parasite. This may be either natural or artificial. Natural active resistance or immunity is derived following repeated exposure to an infection, while artificial active immunity is derived by inoculation of parasite or their stage. It is stronger, longer lasting and diverse and take time to fully develop. An immunity that is developed in host following natural exposure to parasite is actively acquired natural resistance or immunity. Type of natural resistance: different type of natural resistance or immunity wise: pre-immunity, con- concomitant immunity, and sterile or solid immunity are seen in parasitic infection. Pre-immunity. Pre-immunity Preimmunity is a term used to describe resistance that is established after the primary infection has become chronic and is only effective only if the parasite persists in the host, or in other words, preimmunity is immunity developed due to presence of residual parasite in the host. It is also known as incomplete immunity or infection immunity. Confection immunity. Preimmunity is commonly associated with hemoprotozoan disease such as filariasis and babesiosis. Concomitant immunity. Concomitant concomitant immunity. Concomitant immunity, it is one type of preimmunity developed as a result of stimulation of the host is immune system by existing adult parasite. This immunity does not interfere with the survival of existing adult parasite but prevent the entry and establishment of newly invading larvae. This type of immunity is seen in schistosomiasis and cystis echinococcosis, hydatidosis, sterile immunity. Sterile immunity. Immunity maintained by the host even after the parasite is eliminated. The immune system remains sensitized for many years. Theileria annulata infection in cattle, Plasmodium cynomolgi infection in monkey, etc. Self cure. Self cure. Self cure is a phenomena where adult worm populations are expelled following larval challenges. It is a natural remedial mechanism by which the host gains a temporary respite from persist blood loss while aging parasite population is eventually replaced by robust young generation. The expulsion of adult worm population is considered to be a sequel to immediate type hypersensitivity reaction immediate by IgE to antigen derived from developing larvae. Self-cure with not protect the animals in larval challenges often develop to maturity. Self-cure phenomena is commonly associated with Haemonchus contractus infection in small ruminant and cattle. Passive immunity. Passive immunity. Passive immunity refers to transfer of antibodies or lymphocyte from immune to non-immune animal. It is only temporary and does not last long. It could be either natural or artificial. Natural passive immunity refers to transfer of immunity naturally from mother to offspring by placenta or through clostridium, and these antibodies protect the young for a certain period. Artificial passive immunity is immunity transferred to a non-immune animal through injection of serum produced in another animal by repeated injection of particular antigen. The process of producing excess antibodies against pathogen is termed as hyperimmunization. Module 9, Immunity against parasitic infection. Immunity against parasitic infection. Immunity against parasitic infection. Learning objective. The objective of this module is to elucidate the type of immune response against parasitic disease and how the immune response prevents the establishment and development of the parasite in the host beside immunopathological lesion or effect that occurs following an immune response to the parasite. The kind of parasite antigen is also dealt in the chapter. Type of immune response in parasitic disease. Type of immune response in parasitic disease. If the parasite successfully overcomes the animal's innate defense mechanism, it will elicit an immune response specific to the parasite, and these immune responses may be humoral mediated or cell mediated. Humoral immunity is mediated through antibodies, which are large glycoprotein released by B cell. These antibodies are present in blood, lymph, and cerebrospinal fluid. There are five classes of antibodies, each of which is distinguished from the other by their heavy chain. Immunoglobulin G, IgG, major antibody in serum, long half-life. Immunoglobulin M, IgM, largest and the first antibody to be produced in an immune response. Presence denote acute infection. These antibodies are able to neutralize, fix, complement, agglutinate, and immobilize parasitic antigen. Immunoglobulin A, IgA, predominantly found in secretion, mucus, saliva, tears, and colostrum, and hence called secretory antibody. It is also known as mucosal antibody. Immunoglobulin E, IgE, also called as reaging antibody. These antibody bind to the surface of mast cell and cause degranulation of the cell and release of histamine into circulation. This antibody is associated with helminthic infection or allergy. Immunoglobulin D, IgD. The function of this antibody with respect to parasitic infections is not known. Parasites are not composed of a single antigen but a mosaic of antigens. Animal host therefore will recognize a number of these different antigens resulting in a polyclonal antibody response. In addition, the host will produce different immunoglobulin classes having the same antigen specificity. In general, humoral immunity response antibodies control extracellular parasite in the intestine, bloodstream, and tissue fluid. Humoral immune response is predominant in helminthic infection involving IgE, IgA, and IgG antibody. Many helminth infections are associated with characteristic type 1 hypersensitivity, including eosinophilia, edema, asthma, and urticarial dermatitis, where IgE level and eosinophil level are increased. One of the major tasks of eosinophil is the destruction of helminths. Complement. Complement is the name given to a complex of proteins that are present in the serum and which are capable of being activated to become enzymatically active. When complement is attached to an antigen antibody complex, a series of reactions occurs with each component acting enzymatically to active subsequent component in a cascade fashion. Activation of complement can also occur in the absence of immune complex by molecule present on the surface of parasite. How antibody prevent the establishment of parasite? Op- Opsonin. Opsonin are antibody that render the antigen more sensitive for phagocytosis by leukocytes. The presence of complement is essential for opsonization. Ablastine. Ablastine. Ablastines are antibodies that inhibit the reproduction of parasite. These antibodies have been demonstrated in trypanosome infection. They can be transferred from mother to offspring through the milk. Neutralization. Neutralization. Antibody that bind to parasite and neutralize their toxin and inactivate the enzyme. Antibody neutralize the protease used by larvae to penetrate tissue. Antibodies also bind with the excretory and secretory products of helminth and prevent molting. Antibody also act against existing antigen and inhibit larva development. Antibody also block the enzyme pathway of helminthic, resulting in cessation of egg production and development of adult. Immobilization. Immobilization. Immobilization antibody bind to flagella and prevent this parasite from phagocytosis. Lysis of bound parasite with the help of complement. Presence of IgA in secretion will prevent the attachment of parasite in the mucosal surface. Masking of certain target cells by antibodies so that parasites are not able to attach to them. The opening and orifices of parasite are blocked by the antigen antibody complex, causing lysis or death. Cell mediated immunity. Cell mediated immunity. Cellular immunity is mediated by T cell, and like the antibody response, there is involvement of macrophage and the presentation of antigen in combination with specific macrophage surface molecule to the T cell. Specific cytotoxic cell can directly kill parasite infection cell. Some parasite preferentially stimulate T helper cell substrate Th first and Th second, and each helper T cell substance secrete a characteristic set of cytokinin referred to as a T cell cytokinin profile. For example, in Leishmania infection, there will be Th first response with a characteristic cytokinin profile of interleukin, IL-2, IL-3, IL-12, and interferon. Cellular immunity is often directed against intercellular parasite Theileria, Toxoplasma, etc. Immunopathological effect. Immunopathological effect. Immunopathological effect due to immunological reaction against parasitic disease. Immune response evoked by the host against parasitic antigens, causing variety of harmful pathological effect to the host. Pimply gut in esophagostomosis. Esophageal nodule in Spirocerca lupi infection. A vascular granuloma in hepatic cystosomiasis. Hypersensitivity reaction. Hypersensitivity reaction. Hypersensitivity reaction. During the process of destruction and removal of parasite by the host immune system, surrounding body tissue may be damaged. These harmful effects are called as hypersensitivity reaction. The different type of hypersensitivity include type 1 anaphylactic shock. Type 1 immediate reaction due to binding of antigen with IgE fixed to mast cell and basophil leading to release of vasoactive amines, histamine and serotonin. Anaphylactic shock following rupture of hydrated cyst and release of hydrated fluid into the system. Type 1 hypersensitivity is a feature of trichomoniasis and cause local irritation and inflammation in the genital tract. Type 2 cytotoxic is associated with development of anemia in malaria and hemoglobinuria in babesiosis. Type 3 immune complex disease. Circulating antigen antibody complex coupled with complement are deposited in tissue space, causing damage to the tissue. Glomerulonephritis in babesiosis, malaria and schistosomiasis. Type 4 delayed beating fleas secrete saliva into the skin wound and some low molecular weight component present in the flea's saliva act as haptones by binding to dermal collagen, resulting in local type 4 hypersensitivity reaction characterized by mononuclear cell infiltration. This is commonly known as flea bite dermatitis. However, in some sensitized animal, after some month, type 1 reaction take over, resulting in eosinophil infiltration. Kind of parasitic antigen. Kind of parasitic antigen. ES antigen. Antigen from excretion E and secretion S of the parasite are called ES antigen. Somatic antigen. Antigen elaborated by the entire body of the parasite is called somatic antigen. Internal antigen. Antigen obtained after the disruption of the parasite. External antigen manifested by intact parasite. Released antigen metabolic product of parasite are released antigen and these are comparable to external antigen. Concealed or hidden antigen present on the intestinal surface of an mint and arthropod which are not recognized by the host. However, these concealed antigen are excellent target for artificially induced response. Effect of resistance on parasite. Prevention or reduction in the rate of establishment, structure or metabolic alteration in the parasite. Retardation or inhibition of development. For example, adult female osteotegii recovered from immunized calves show absence of vulvar slab as compared to those recovered from non-immunized calves. Similarly, Coperia male derived from immunized animals show altered morphology of spicules. Inhibition of reproduction, for example, in ticks that feed on immunized animal, there will be Extended evaporation reduce egg laying and diminished hatchability of egg. Destruction of parasite in situ elimination of existing infection. Thank you.